ever get that feeling like you're drowning in information? Especially when it comes to something like rare diseases, you know? It can be so overwhelming trying to figure out what's going on, like where do you even start? You're looking for a diagnosis, but it feels like you're lost in a maze, blindfolded.
Oh, absolutely.
And what if I told you there's a whole new way to approach this? A way to look at the bigger picture, the whole language of our cells, for clues.
That's a really intriguing thought. And actually, that's exactly what we're diving deep into today.
Yeah, we're talking about a totally new approach to diagnosing these rare diseases. And instead of getting lost in the weeds of individual genes, we're going to zoom out and look for these broader patterns that might be pointing us towards the problem. So to get started, we're going to be looking at a really cool preprint study.
Okay.
Now remember, this means it hasn't gone through the whole peer review process yet.
Right.
But the findings are super exciting. This study comes from the Gregor and the UDN, which are the genomics research to elucidate the genetics of rare diseases, and the Undiagnosed Diseases Network.
All right, the big leagues.
Yeah, exactly. And they looked at a pretty decent-sized group, 390 individuals.
Yeah, it's a good cohort.
Using this really cutting-edge technique called RNA sequencing. RNA-seq?
RNA-seq, yeah.
Now, we all know DNA is like the instruction manual for our bodies.
The blueprint.
The blueprint, yeah.
Right.
But RNA, that's the messenger.
It's the courier.
Yeah, it's the one that carries the instructions from the DNA to build the proteins, which are the ones actually doing all the work in our cells.
The workers, yeah.
The workers, yeah. So what's so cool about this new method is that it looks at how those RNA messages get put together.
Like the assembly line?
Yeah, exactly. The assembly line. And that process is called splicing.
Splicing, that's a key step.
So splicing, you know, for those of us who haven't spent our lives in a genetics lab,
right,
it's basically like editing a really, really, really long document.
I like that analogy.
So think of a gene as a recipe, right? But it's got a bunch of extra steps thrown in that you don't really need.
It's fluff, yeah.
Yeah, fluff. So splicing is like that super meticulous editor who comes in and cuts out all the unnecessary stuff. Those are called introns.
Okay.
And then they carefully piece together the important parts, the exons, to make that final, concise set of instructions to build a protein.
So it's like making sure the recipe is clear and makes sense.
Yeah, exactly. And as you can imagine, if that editing process gets messed up...
Oh, disaster.
Yeah, you could end up with a protein that doesn't work right.
Or doesn't work at all.
Yeah, and that can lead to all sorts of problems, ultimately disease.
Absolutely.
Now, traditionally, when doctors try to figure out what's causing a genetic disease, they've usually focused on finding these tiny mutations in a single gene.
Like looking for a typo.
Yeah, like one single typo in that massive recipe book.
Right, right.
But what if the problem isn't in the gene itself, but with the machinery that's supposed to be doing the splicing?
That's a good point, yeah.
Like, what if the editor is the one making the mistakes?
That would be a big problem.
Yeah, a big problem. And that's where this whole transcriptomics-first method comes in.
Okay.
It's looking for signs of trouble in the splicing process, but not just in one gene, but across many genes.
So instead of zeroing in on one specific spot, we're taking a wider view.
Yeah, we're looking for a pattern of errors that could point to a problem with the splicing machinery itself. So to get even deeper into this, we need to talk about the actual players involved in the splicing action.
Okay, the splicing team.
Yeah. So it turns out our cells don't just have one splicing team.
Oh.
They actually have two.
Two teams.
Two main teams. There's the major spliceosome.
Okay.
Which is kind of like the workhorse.
Right.
It handles the vast majority of the splicing, like 99.5%.
Wow, that's most of it.
Yeah. But then we have this more specialized team, the minor spliceosome.
Okay.
And they deal with the remaining tiny fraction, like 0.5% of those introns.
The minority, but still important.
Right. And while that might sound small, it actually involves around 800 specific regions in our genes.
Okay.
Which we call minor introns.
Minor introns, got it.
And guess what? Today's deep dive is all about this often overlooked minor spliceosome.
Interesting. So we're focusing on the specialist team today.
Exactly, because understanding how this minor spliceosome works, and especially how it can go wrong, could be the key to diagnosing a whole bunch of rare diseases.
I see. So the goal of our deep dive is to figure out how looking at these transcriptome-wide splicing patterns, and particularly in those genes with the minor introns, the MIGs, can help us crack the code of these challenging diseases.
Exactly. And maybe even point us towards new genes that we didn't even know were involved.
Right. New culprits.
New culprits.
Yeah.
Okay, so let's start with the basics. What was wrong with the old way of doing things? Why wasn't focusing on single genes always enough to find a diagnosis?
Well, think about it this way. When we just look for outliers in a single gene, we're assuming that the problem is limited to that one gene.
Like a spotlight.
Yes, like we're shining a spotlight on just one spot.
Right.
But if the issue lies in the splicing machinery itself,
Right.
that affects so many genes.
Yeah.
It's like we're missing the whole forest for the one tree.
Okay, so it's like if you're trying to fix a computer and you're just looking at one program.
Right.
But the problem is actually with the operating system.
Exactly. You could spend forever trying to fix that one program and never solve the real problem.
So the spliceosome itself, it's not just one simple thing.
No, it's actually quite complex.
It's got over 300 different protein parts.
Wow, that's a lot of moving parts.
Yeah. Plus those two snRNA machineries we talked about, the major and the minor.
Right.
So if there's a defect in any of those components, it could mess up the splicing of tons of genes, not just one.
Right, like a domino effect.
Yeah, and there's been research showing that this is actually the case. That problems with the spliceosome can lead to these widespread splicing errors across the whole transcriptome.
So the traditional approach might have been too narrow, too focused.
Yeah, we were missing the bigger picture.
Missing the forest for the trees.
Okay, so how does this new transcriptomics-first method change things? How does it work?
Well, instead of starting with a specific gene,
right,
we start by looking at the RNA-seq data for any signs of too many splicing errors.
Across the whole transcriptome.
Exactly. We're casting a wide net.
Okay, so they're basically looking for people whose RNA is just riddled with splicing glitches.
Yeah, more than you'd expect to see normally.
And how did they actually do this in their study? Like, how do you even find those splicing errors?
They use these really sophisticated tools called Fraser and Fraser 2.
Okay.
And these tools are specifically designed to pick out these abnormal splicing events in the RNA-seq data.
So like a spell checker for RNA.
Kind of, yeah. They're looking for those mistakes in the splicing process.
And in this study, they were particularly interested in one specific type of error called intron retention.
Intron retention, yeah.
Now remember, introns are those parts of the gene that should be removed during splicing.
The fluff?
The fluff, yeah. But in intron retention, those introns are left in the final RNA message.
So it's like the editor missed a step.
Yeah, they left in some of the unnecessary sentences.
And that can really mess things up.
Totally, because now you've got this garbled message that might not be able to make a functional protein.
Right. It's like trying to bake a cake with a recipe that still has the instructions for making the frosting mixed in.
Exactly. And because they were focusing on the minor spliceosome, they specifically looked for intron retention errors in MIGs.
Those minor intron-containing genes.
Yeah, because if the minor spliceosome is the one having problems,
you'd expect to see more errors in the genes it's supposed to be handling.
Right. Like if the specialized editor is messing up.
You'd expect to see more problems in the specialized documents.
Okay, so they're looking for these people with lots of intron retention outliers, specifically in MIGs. What did they find?
Well, the exciting part is they found 5 individuals.
5?
5 individuals who had a way higher number of these errors than everyone else.
Wow.
And when they dug deeper into their genetics, they found that all 5 had these rare mutations in genes that are responsible for making snRNAs.
snRNAs.
Yeah, small nuclear RNAs.
Yeah.
And these snRNAs, they're like the building blocks of the splicing machinery.
Oh, so they're like the tools the editors use.
Exactly. So these 5 individuals, they had mutations in the genes that make the tools for splicing.
So it's not just that their genes were messed up.
Right.
It's that the tools for editing the genes were messed up.
Exactly. Which means you're going to see problems across lots of different genes.
Wow, so it's like a fundamental problem with the whole editing process.
Absolutely. And for these 5 individuals, they had these specific mutations in a gene called RNU4-ATAC.
RNU4-ATAC, okay. And these mutations were compound heterozygous?
Yes. That means they inherited a different messed up copy from each parent.
Like 2 different typos in the same sentence.
Yeah, but on different words. And you know what's interesting? Finding these mutations actually helped clarify some previous uncertainties about this gene.
Oh, really? So they were able to reclassify some variants that they weren't sure about before?
Yeah, 4 of them. Yeah. Just by finding these individuals with clear-cut mutations.
Wow. So this method is not just about finding new things, it's also about refining our understanding of what we already know.
Absolutely. It's like cleaning up the dictionary.
Okay, so let's talk more about this RNU4-ATAC gene. What does it actually do?
So RNU4-ATAC is the blueprint for this little RNA molecule called the U4-ATAC.
U4-TAC.
Yeah, and U4-TAC, that's a crucial part of the minor spliceosome.
So that's like a key player in that specialized editing team.
Exactly. It helps the minor spliceosome recognize and cut out those minor introns.
Right, the fluff and the specialized documents.
Yeah, and this U4-TAC, it's got all these important parts.
Oh, like stem I, stem 2, and these little loop structures.
Like different parts of the editing tool.
Right, and each part has a specific job to do.
Makes sense.
There's even a special spot where it binds to these enema MAPO proteins.
Sim proteins?
Yeah, these proteins are like the glue that holds U4-ATAC together.
Okay.
And make sure it's stable and working properly.
So they're kind of like the tool maintenance crew.
Yeah, I like that analogy.
So back to the 4 individuals with the RNU4-ATAC mutations. Where were those mutations located and what kind of impact could they have?
Well, the researchers were really meticulous about this.
Okay.
They mapped out exactly where these mutations were on the U4-ATAC molecule.
Like highlighting the typos.
Yes, exactly. And they found some interesting things. For example, individual A1,
right?
They had one mutation in stem 2.
Stem 2, yeah.
Which is known to be super important for splicing.
Okay.
And another mutation in the region where it binds to those enema proteins.
The tool maintenance area.
Yeah, and that region can be important too.
Then you have individual B1.
Yeah.
They had mutations in both stem 2 and the STIRSCOM protein binding region.
Double whammy.
Yeah, and both of those mutations were in areas that are thought to be really crucial.
So likely to have a big impact.
Yeah. Then there were siblings, C1 and C2.
Okay.
They shared one mutation in the SUM protein binding region.
Okay.
And another in this 5-stem loop, which is a little less clear how important it is.
Interesting.
So even though they're all related to RNU4-ATAC,
right,
the specific mutations can be quite different.
And potentially affect different parts of the splicing process.
Exactly. Which is why you can see such a range of symptoms even with the same underlying gene problem.
Like with those Swiss watches, even a tiny problem can throw everything off.
Exactly. It's all about precision.
So did these different mutations correlate with how these individuals were actually affected, like their symptoms?
That's the amazing part, yes. All 4 individuals had clinical features that were consistent with what's known as RNU4-ATACopathy.
RNU4-ATACopathy, so that's a group of conditions related to RNU4-ATAC problems.
Exactly. And these conditions, they've historically been known by different names, like MOPD1, Roifman syndrome, and Lowry-Wood syndrome.
Wow, a lot of names for the same underlying problem.
Yeah, it just goes to show how complex this all is.
So what kind of symptoms are we talking about here?
Some of the common features include growth restriction, microcephaly, which means a smaller head size, skeletal dysplasia, problems with bone development, and intellectual disability.
Okay, so some pretty serious developmental issues.
Yeah, unfortunately. But they also found some less common features in these individuals.
Oh, really?
Like immunodeficiency, problems with the immune system, and issues affecting the heart, eyes, and endocrine system.
So it really highlights the range of ways this gene can affect people.
Absolutely. And the key finding here is that the number of MIGs...
Those minor intron-containing genes?
Yes, the number of MIGs with intron retention errors in these 4 individuals. It was significantly higher than in the rest of the study group.
That really solidifies the link between the gene and the splicing errors.
Exactly. It's like seeing the dominoes fall.
Okay, so we've got 4 individuals with RNU4-ATAC mutations, but there was that 5th individual, right? The one without mutations in RNU4-ATAC or RNU12.
Yes, and this is where it gets even more exciting.
Oh.
This individual, D1, also had a ton of intron retention errors in MIGs.
So the same basic pattern as the others.
Exactly. Yeah. But without the usual suspects.
So what did they do?
They did a more targeted analysis. Looking specifically at other genes involved in the minor spliceosome.
Like narrowing down the suspects.
Yeah. And they hit the jackpot.
They found something.
They found rare compound heterozygous mutations.
Again, with the 2 different mutations.
Yeah, but this time in a gene called RNU6-ATAC.
RNU6-ATAC, that's a new one.
It is. And it's potentially a brand new disease gene.
So they stumbled upon a whole new piece of the puzzle.
Absolutely.
Yeah.
And this opens up a whole new avenue for research.
That's huge. So tell me more about this RNU6-ATAC gene. What's its role?
Well, like RNU4-ATAC, it also makes an RNA molecule. Called U6-TAC.
U6-TAC?
Yeah, and the 2 mutations they found in individual D1, they were super rare.
Like almost unheard of.
Yeah, they checked this massive database called NOMAD, and hardly anyone else had these mutations.
Wow, that's pretty compelling evidence right there.
And get this, these mutations were in a super conserved part of the gene.
Conserved, meaning it's stayed pretty much the same across different species.
Exactly. They looked at the Phylop scores.
Phylop scores.
Yeah, it's a way to measure how much a particular DNA or RNA sequence has changed over millions of years.
Okay.
And these scores were really high, like 6.99 and 7.69.
Wow.
Which means that this region of the gene is really important. Evolution hasn't messed with it much.
Because if you mess with it, things probably go wrong.
Exactly.
Yeah.
So these weren't just random changes. They were in a very sensitive spot.
Okay, so these mutations are likely to have some serious consequences. What does this U6-ATAC actually do?
Well, U6-ATAC is super important because it interacts directly with U4-ATAC.
U4-ATAC, the one from the other gene.
Yeah, they have this special bond called base pairing.
Base pairing, that's where the nucleotides match up.
Exactly. And this interaction is essential for forming the catalytic core of the minor spliceosome.
The catalytic core, that's the part that actually does the cutting.
Yeah, it's called the tri-SNRNP.
Tri-SNRNP.
Okay. It's like U4-ATAC and U6-ATAC come together like two puzzle pieces to create this cutting tool.
And if one of the pieces is misshapen,
the tool doesn't work right.
So could those specific mutations in RNU6-ATAC be messing up that interaction with U4-ATAC?
That's what the researchers think.
Okay.
They pointed out that one mutation, the N.28CT1, could be affecting how U6-ATAC binds to a specific part of U4-ATAC called STEM-I.
STEM-I, we talked about that. It's important.
Yeah, and then the other mutation, the N.36CT1.
Yeah.
That could be destabilizing the whole U6-ATAC structure.
Like making the puzzle piece wobbly.
Yeah. Or it could be messing with how it binds to STEM2 of U4-ATAC.
So either way, U4-ATAC and U6-ATAC can't connect properly.
And the whole minor spliceosome gets thrown off.
Okay, so now let's talk about this individual D1. What were their symptoms like?
This is another really interesting part. Individual D1 had growth restriction.
Okay.
Microcephaly, epilepsy.
Epilepsy, wow.
Intellectual disability, and ataxia.
Ataxia, that's problems with coordination, right?
Yeah, exactly.
So those are all pretty similar to the symptoms we saw in the RNU4-ATAC cases.
Yes, there's a significant overlap.
So even though the mutations are in different genes, the end result is kind of similar.
And that's because both genes are involved in the same process, the minor spliceosome.
Okay, so we've got similar symptoms and the same basic pattern of splicing errors.
Right.
Which strongly suggests that RNU6-ATAC could be a new disease gene.
A very strong candidate.
Yeah.
And to make it even more convincing,
yeah.
they found that 141 out of the 153 genes with intron retention errors in individual D1 were also messed up in the RNU4-ATAC cases.
So like 92% overlap.
Yeah, that's a lot. It's really pointing towards a shared underlying mechanism.
So this transcriptomics-first approach basically led them to a whole new potential disease gene.
Yeah, it's a really powerful tool for discovery.
But you keep saying candidate gene. What else needs to happen to confirm that this is really a disease-causing gene?
You're right. We need more evidence. Ideally, we'd find more individuals with similar mutations in RNU6-ATAC.
Right.
And see if they have similar symptoms.
Like replicating the findings.
Exactly. And we'd also want to do some lab experiments.
Okay.
To see how these specific mutations actually affect the minor spliceosome.
So like take the mutated gene and see how it behaves in a cell.
Exactly. That would give us even more direct evidence.
Okay, so more research is definitely needed. The study also mentioned some interesting things about sample size and batch effects. Can you explain those?
Yeah, so sample size, that's just the number of people in the study.
Right.
And they found that the more people they included, the more intron retention outliers they found in MIGs.
So bigger studies are better?
It seems like it, yeah. For example, they looked at one of the RNU4-ATAC cases.
Okay.
And when they analyzed their data in a larger data set,
yeah.
they found way more errors than when they just looked at that individual's data alone.
So it's like having a wider lens.
Exactly. You can see more detail.
Makes sense. And what about those batch effects?
That's a tricky one. Batch effects are basically variations that can happen when samples are processed differently.
Like if one lab does things slightly differently from another lab.
Yeah, or even if you process samples on different days using slightly different equipment or chemicals.
Right.
And these variations, they can really mess with your results.
Oh, no.
Yeah, the study found that batch effects could significantly affect the number of splicing outliers they detected.
So it's like adding noise to the data?
Yeah, like static on a radio signal.
Yeah.
So it's really important to correct for these batch effects in future studies.
So you can be sure that the differences you're seeing are real.
and not just due to technical issues.
But did they say anything about whether batch effects were a problem in their own study?
They did. They said that even though their samples were processed in different ways, the specific signature they were looking for,
the increased intron retention in MIGs,
that signal was still strong and statistically significant.
So their key findings weren't just a result of batch effects.
Exactly. That's good to know.
They also mention comparing their approach to another method called Fraser-2, using something called the Jacquard index. What was that about?
Yeah, so Fraser-2 is another tool for analyzing RNA-seq data.
Okay.
And they found that it could also identify those 5 individuals with the minor splicing problems.
Okay.
But it wasn't as specific.
What do you mean?
It also flagged 2 other individuals who didn't have any known mutations in minor spliceosome genes.
So it might have been picking up on some things that weren't actually related?
Yeah, like false positives.
So their original method seems to be more accurate.
More focused, yeah.
Okay, so we've covered a lot of ground here. What's the big takeaway from all of this? What should we as listeners be thinking about?
I think the main message is that this study shows us a whole new way to think about diagnosing rare diseases.
Okay.
Instead of getting bogged down in individual genes,
right,
we can look for these broader patterns of splicing errors.
Especially in those minor introns.
Yeah, and this can help us find diagnoses for people who might have been searching for answers for years.
And it can also lead to the discovery of new disease genes, like HARNU6A TAC.
Exactly. It's opening up new possibilities for diagnosis and treatment.
It's like shifting from looking at individual trees to seeing the whole forest.
Yeah, and realizing that the health of the forest depends on so many interconnected things.
And who knows what other patterns are out there waiting to be discovered.
That's the exciting part. There's so much more to learn.
It really makes you think about the bigger picture, the complexity of our biology.
Yeah, it's amazing how much is going on inside ourselves.
And how even tiny errors can have such big consequences.
But now we have new tools to help us understand those errors.
And hopefully find ways to fix them.
Absolutely. That's the ultimate goal.
Well, this has been a fascinating deep dive. Thanks for taking this journey with me. It's really given me a lot to think about.
Yeah, it's been a pleasure exploring this with you.
It's amazing how much progress is being made in this field.
Yeah, it's a really exciting time for rare disease research.
I can't wait to see what they discover next.
I'm sure there will be more breakthroughs just around the corner.
Thanks again for joining me.
My pleasure.
And to all our listeners out there, keep those brains buzzing.
Yeah, stay curious.
And until next time, happy splicing.
Happy splicing.